Welcome back to Parasitology. Hi, I'm Christopher. In this video, we are entering the flagellate lane, motile protozoa that may share a general way of moving, but certainly do not share the same clinical address. We will cover some of the major flagellates: Giardia, Tritrichomonas, Trichomonas, and Histomonas. Sort each one by 4 clues: the host, the anatomic site, the important life cycle stage, and the syndrome. Same lane, very different neighborhoods. By the end, a puppy, a young cat, a pigeon, or a turkey should point you towards a very different flagellate pattern. Here is the whole lesson in map form. The first lane contains the diplomonids. Giardia duodenalis, also called Giardia intestinalis, is the most familiar clinical example. It infects a wide range of mammals, including dogs, cats, livestock, rodents, wildlife, and even marine mammals such as seals. The basic pattern is the same: the small intestine, cysts and trophozoites, and diarrhea, although infection does not always produce clinical disease. Spironucleus and hexamida provide some useful aquatic animal context, but they are not our main focus today. The second lane contains the trichomonids and related flagellates. Trichomonas foetus should make you think of a young cat with chronic large bowel diarrhea. Trichomonas gallinae should make you think of a pigeon, dove, or raptor with oral or crop lesions. Histomonas meleagridis should make you think of a turkey, the ceca and liver, and blackhead disease, often with heterachus and earthworm involvement. Now notice the stage pattern. With Giardia, both cysts and trophozoites matter. The others are primarily discussed as trophozoites. Remembering the difference will keep you from trying to dress every flagellate in Giardia's life cycle wardrobe. Before we zoom in, place these organisms on the larger parasitology map. They are eukaryotes, just like their animal hosts, and inconveniently, just like us. Parasitology is therefore a very distant form of family drama. The word flagellate is the most useful here as a practical description rather than a single tidy taxonomic box. Giardia and Spironucleus or Hexamita are diplomonids within the broad excavatin portion of this map. Tritrichomonas and Trichomonas occupy a different branch, and Histomonas is another related flagellated protist. Taxonomic frameworks continue to evolve, but your clinical sorting rule remains stable. Identify the host, find the home, recognize the stage, and connect the syndrome. The taxonomy tells us that these organisms are not all close cousins. The clinical pattern tells us why that matters. Now compare the organisms side by side. A trophozoite is the motile feeding stage. The term comes from the Greek root associated with nourishment and a living organism, which is a fairly accurate job description. This is the stage actively living and feeding in the host. In this teaching set, Giardia is the cyst-forming standout. Its trophozoites live in the small intestine, while the environmentally resistant cyst is the major infective stage. Trichomonas foetus is the trophozoite focused in the feline large intestine. Trichomonas gallinae is the trophozoite in the oral cavity and crops of birds. Histomonas meleagridis is associated with the turkey, ceca, and liver, and transmission is tied to the heterachus eggs and sometimes earthworms. Spironucleus and hexamida infect fish and amphibians, and the practical message is simple. Do not diagnose from the word flagellate. Diagnose from the combination of host, site, stage, transmission pattern, and clinical clue. The word flagellum comes from Latin for little whip, and Giardia carries 8 of them, arranged as 4 pairs. Those flagella help create the movement that makes fresh trophozoite so recognizable. Trophozoite is bilaterally symmetric and has 2 nuclei, producing the famous face-like appearance. It is not actually looking back at you, although under the microscope it can feel mildly judgmental. On the ventral surface is an adhesive disc that helps the trophozoite attach to the small intestinal epithelium. Basal bodies organize the flagella, and the median bodies provide internal structural support. The cyst is the durable stage. Its protective wall permits environmental survival, and a mature cyst can contain up to 4 nuclei. Trophozoites attach and multiply in the small intestine. Cysts are fed in the feces and serve as the major infective form and an important diagnostic target. For this lane, remember the contrast. Giardia gives you trophozoites plus cysts. The remaining organisms are trophozoite-focused. Tritrichomonas fetus is the young cat large bowel flagellate. The name contains Greek roots for 3 and hair, reflecting the prominent hair-like flagella associated with this organism. Clinically, think of a kitten or young cat, especially from a dense multi-cat environment with chronic or recurrent large bowel diarrhea. The feces may be malodorous, contain mucus, and tenesmus may occur. The organism lives in the large intestine colon, not the small intestine. It is also trophozoite-focused without Giardia's central environmental cyst stage. On a fresh fecal wet mound, the trophozoites are elongated and motile, often with jerky, wavy movement created by the flagella and undulating membrane. Because these organisms are fragile, fresh samples should be examined promptly. Culture systems and PCR provide additional diagnostic context. The memory equation is young cat, chronic large bowel diarrhea, and a motile tritrichomonid equals Tritrichomonas fetus. Giardia lives at a different intestinal address and brings the cyst. Before we leave this organism, one nomenclature note. The feline trichomonid is still commonly called Tritrichomonas fetus, although some sources use the name Tritrichomonas blackburni. Its status as a separate species remains debated. Feline and bovine isolates are closely related, but they are not genetically identical and demonstrate important biologic and host-associated differences. Tritrichomonas blackburni was proposed as a separate species for the feline organism, although that classification has not been universally adopted. For practical clinical sorting, think of these as 2 closely related, host-adapted clinical lanes, rather than assuming one interchangeable parasite simply moves between cats and cattle. In the bovine setting, Tritrichomonas fetus is a venereal transmitted reproductive pathogen. Bulls may carry the organism in the preputial cavity without obvious signs of clinical disease and transmit it during breeding. In cows, the important clues include reduced conception, early embryonic loss, and occasionally abortion. Diagnosis therefore requires reproductive or preputial sampling rather than a feline fecal sample. Now move from the feline colon to the avian oral cavity. Trichomonas gallinae is associated primarily with pigeons and doves, but raptors may become infected after eating infected prey. The species name gallinae comes from the Latin gallina, meaning hen or chicken, which is a useful hint that we have entered the bird lane. The primary sites are the oral cavity, pharynx, crop, and sometimes the upper gastrointestinal tract. The classic clue is a caseous oral plaque. In pigeons and doves, the disease is commonly called canker. In raptors, it is often called frounce, a traditional falconry term for trichomoniasis involving the mouth, pharynx, or crop. Affected birds may develop dysphagia, weight loss, and weakness. Transmission can occur through saliva, regurgitated feedings of nestlings, and shared contaminated food or water. For raptors, lunch quite literally comes with consequences. Diagnosis may involve an oral swab or crop sample with a fresh wet mount used to look for motile trophozoites. Unfortunately for many, diagnosis may also occur at necropsy. Remember, bird plus oral plaques plus motile trophozoites point towards Trichomonas gallinae. Think canker or frounce, not diarrhea. Histomonas meleagridis completes our avian trouble. The species name meleagridis refers to meleagris, the turkey genus, so the name itself points us towards the most important host. The primary site is the ceca, with the classic lesion pattern involving both the ceca and liver. Affected turkeys may show depression, poor growth, sulfur yellow droppings, and potentially high mortality. At necropsy, the mental image is necrotizing typhlitis with cecal cores and characteristic target-like liver lesions. The transmission story is what makes this organism so memorable. Histomonas is linked to the eggs of the cecal worm Heterachus gallinarium. Earthworms may serve as transport hosts carrying infected Heterachus stages into the turkey's environment. In other words, think of a parasite traveling in a worm ending up in a turkey. The slide calls this the turducken of death, and unfortunately the biology earns the nickname. Your pattern is turkey, ceca and liver, Heterachus or earthworm context, and blackhead disease. This should make you thinking Histomonas meleagridis. Let's finish by running the 4 major patterns one more time. A puppy or dog with small intestinal diarrhea, especially when cysts are part of the picture, points towards Giardia. Don't forget that Giardia also affects horses, ruminants, rodents, and seals. A young cat with chronic large bowel diarrhea and a motile trophozoite-focused organism points towards Tritrichomonas foetus. A pigeon, dove, or raptor with oral or crop plaques, canker, or flounce points towards Trichomonas gallinae. A turkey with cecal and liver disease, blackhead, and Heterachus or earthworm context points towards Histomonas meleagridis. Do not begin with the organism name, begin with the patient. Sort by host, site, stage, and syndrome, and the organism becomes much easier to retrieve. Puppy, young cat, pigeon, turkey. 4 hosts, 4 homes, 4 very different clinical clues. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.